everyone. Today we are going to discuss Lesson 11, the light-independent reaction of photosynthesis, also known as the Calvin cycle. So, for our objectives today, first we have to describe the significant events of the Calvin cycle, detail the three steps of the light-independent reactions, and define carbon fixation. Before we proceed to our lesson today, let's have a short recap on our previous lesson about the light-dependent reaction because what we are going to discuss today is a continuation of our previous lesson. Within plant cells, chloroplasts are specialized organelles that serve as the sites of photosynthesis. The reactions that make up the process of photosynthesis can be divided into light-dependent reactions, which takes place in the thylakoid, and light-independent reactions, which take place in the stroma. Light-dependent reactions harness energy from the sun to produce chemical bonds, ATP and NADPH. These energy-carrying molecules are made in the stroma where the carbon fixation takes place as part of the light-independent reactions or the Calvin cycle. The entire image represents the chloroplast, which is bound by a double membrane. The solution within the chloroplast is the stroma. A portion of the thylakoid membrane is on the left and encloses the thylakoid's lumen. Embedded in it are various photosystems and protein complexes which form an electron transport chain. Excited electrons move along the transport chain and are ultimately used to reduce NADP+, producing NADPH. The energy from electrons is used to pump protons into the thylakoid lumen. Protons move down their concentration gradient through ATP synthase, generating ATP. Water is oxidized to replace lost electrons, releasing gaseous oxygen. Okay, so now let us move into light-independent reaction, better known as the Calvin cycle. The leaves of this plant have cells that carry out photosynthesis. If we zoom in on this photosynthetic plant cell, we can see the chloroplast where the reactions of photosynthesis occur. Photosynthesis consists of two primary steps, the light-dependent reactions and the Calvin cycle reactions. In this video, we'll focus exclusively on the Calvin cycle reactions. The overall purpose of Calvin cycle is to convert carbon dioxide from the atmosphere into carbohydrates or sugar, which the plant needs to power its cellular activities and build new plant structure. The Calvin cycle reactions occur within the stroma of the chloroplast. The reaction uses the ATP and NADPH produced by the light reaction to convert the carbon dioxide to carbohydrates. Let's zoom into Calvin cycle to take a closer look. The Calvin cycle can be divided into three phases: carbon dioxide fixation, carbon dioxide reduction, and regeneration of RUBP. The first step is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-biphosphate, resulting in a 6-carbon molecule that splits into 3 carbon molecules. The second step is sequence of reaction using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the third step, Ribulose 5-biphosphate is regenerated for every step of the cycle. 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-biphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. And that's all for our lesson today. Thank you guys for watching.